Antibodies are a group of proteins with a specific immune function. Under normal circumstances, these molecules, which are produced by white blood cells called lymphocytes, recognize and bind to complementary proteins known as antigens on the surface of invasive pathogens. In doing so, they prevent these pathogens from causing further damage to the body. However, antibodies' unique specificity also makes them valuable as a diagnostic tool. We define monoclonal antibodies as identical collections of antibodies, all of which have been produced by a single lymphocyte. The fact that these molecules all possess identical binding sites allow them to be used to detect elevated hormone levels during pregnancy, to deliver medicines to specific tissues, and to treat individuals for certain diseases. At Key Stage 4, students must be able to recall the following steps by which monoclonal antibodies are produced. First, scientists inject a specific antigen into a host mouse. In response, the mouse's immune system generates specific lymphocytes which themselves produce a vast reservoir of antibodies specific to the target antigen. The scientists then perform a small operation on the mouse, removing spleen cells which contain a high density of these lymphocytes. These cells are fused with cancerous white blood cells taken from a human called myeloma cells. The resulting cell is known as a hybridoma and can divide indefinitely. As the hybridoma cells divide, they produce millions of specific monoclonal antibodies which can be harvested and utilized for medical or commercial purposes. It's crucial that students appreciate the potential power of monoclonal antibodies, but understand that any advantages must be balanced with the ethical implications of animal-based production and testing. The fact that host mice are often killed during synthesis means that advances in the area have been slow, and many long-term implications remain unclear.